Good day to all of you. Here is another video regarding the topic enzyme, their classification and nomenclature. So enzyme are the biocatalyst which increases the rate of reaction without getting consumed in this overall process. So it lowers the activation energy, and what is activation energy we will see in the later stages. So the term enzyme was coined by de Kuhne, and in properties of enzymes are as follows. Like the enzymes have several properties that they make them the unique, and these are the mostly enzymes are proteinaceous in nature. They are highly specific for their substrate and the rate of reaction by lowering the activation energy. It doesn't change the equilibrium state of the biochemical reaction, but only changes the rate of at which equilibrium is achieved. So what is activation energy? It is the minimum energy required to change the substrate from the stable state to the transition state. So here is a graph of the activation energy with enzyme, activation without enzyme. So enzymes decreases or the lowers down the activation energy. So enzymes act as a tertiary structure level which provides many services or the pockets like structure called the active sites, and which are substrate specific. So here the classification and the types of the enzymes. So enzymes contains RNA, protein, and the protein may be the simple enzyme and the conjugated enzyme. Simple enzyme contain only proteins, while the conjugated contains proteinaceous plus non-proteinaceous part. The cofactor is called as the non-proteinaceous part, and the proteinaceous part is called as the apoenzyme. The non-proteinaceous, that cofactor, is maybe the organic cofactor or inorganic cofactor. What is prosthetic group? It is firmly or the covalently attached with the apoenzyme known as a prosthetic group. That is example heme for the enzyme peroxidase catalase. Coenzyme is loosely attached or they attached only at the time of the reaction. And metals are generally some metals ions which act as a cofactor like metal activated group and the metalloenzymes. So exoenzymes is an intracellular enzyme which are produced within cells and act outside the cell, while the endoenzyme is an intracellular enzyme which are enzymes which produce within the cell and act within the same cell. So here is another subtopic, that is models of enzyme. The two important models of enzyme, that first one is the lock and key theory by Emil Fischer, and second one is the induced fit theory given by the Daniel Kosland. So in this theory, according to this theory, the active site of an enzyme is prefixed and is very similar to the geometrical shape of the substrate. So here is the figure of the lock and key model, that is the active site. These are the active site of an enzyme which is substrate specific and is similar to the shape of the substrate. Induced fit theory says that the active site of an enzyme is not prefixed, rather than with induced to attain the shape similar to the substrate when substrate attached with the enzyme. An active site has two components, that is buttressing site, and the second one is the binding site or the catalytic site. So in this enzyme, it is the binding site and it is the buttressing site, and substrates are there, and when it attached with this, it forms the complex enzyme product complex. So binding of S1 in enzyme binding site leads to the conformational changes in enzyme and bond formation between the substrate 1 and substrate 2. So next is the classification of the enzyme. So on the basis of type of reaction, it may be classified into 6 classes. So we can remember it in this word like over the hill, OTHILL. So oxo-reductase enzyme catalyze the oxido-reduction between the two substrate in order to form a product. So in this figure, A is in the reduction form and B is in oxidized form. And when reaction happens, A becomes oxidized and B becomes reduced. So transferase. These enzymes facilitate the transfer of the functional group between the substrate in order to form a product. Hydrolysis enzymes breaks down the substrate molecule by using water and in order to make them a product. And in isomerase, it facilitates the intramolecular changes in the substrate, like interconversion of the optical, geometrical, or the positional isomers. Lyase enzyme, it catalyzes the non-hydrolytic removal of the group to the substrate in order to make them a product and leaving a double bond. Ligase enzymes catalyzing the linking together or joining of two substrates by forming a new bond. Next is the factors affecting the enzyme activity. First one is the temperature. So optimum temperature is required for the reaction process, and if it decreases or increases, the enzyme activity will decrease the reaction. So here is a graph of the temperature and the rate of reaction. So it forms a bell-shaped curve that is a graph of the temperature, effects of temperature on enzyme activity. So next is the pH level. So it is same as the temperature. Enzyme will decrease when the pH will increase, either decrease or increase. So here is the graph of the rate of reaction and the pH. It also forms the bell-shaped curve. Next is the substrate concentration. So the substrate concentration, the initial increase in substrate concentration will increase the reaction, but after the saturation of the active site, then there will be no change in the rate of reaction, and it forms a rectangular hyperbola graph. So next is the product formation. Increase in product concentration will decrease the enzyme activity. Last one is the presence of the inhibitors or the activators. Inhibitor binds to the enzyme and present the formation of the enzyme substrate complex, and thus inhibits the reaction. And if the concentration of the inhibitors are more than the substrate, then no reaction takes place. So in this figure, if the inhibitors binds to the active site of an enzyme, there will be no reaction. So when activator binds to the enzyme, a conformational changes occur in the active site of an reaction takes place. So this is all about the classification, nomenclature, their types, and the factors affecting the enzymes. So thank you.